Okay, assalamu alaikum. So let's go over the second topic of the molecular biology part of biochemistry. And the focus of this topic is on enzyme-based molecular techniques. Okay. Again, resource this lecture, and you can refer to Cooper's. So we will start talking about DNA sequencing. Okay, so that's the first. Now, what is DNA sequencing? It's basically knowing the order of nucleotides in a piece of DNA or a whole genome, maybe. So you know that the starting point, let's say it's A, C, G, T, A, G, C, T, etc. Okay, all the way to the end. It's almost like reading the letters in a book. Like for example, THEBOKISNICA. Okay, it's meaningless unless you translate or you know what the code is to change these letters into meaningful words. So the book is nice. So this is what DNA sequencing is, and by knowing the DNA sequence, you can tell a lot of things. One, you can identify genes and their localization. So the idea is that you can identify words and where they are located in a book. Okay, so you can pinpoint that this is a sequence of nucleotides that represent the beginning of a gene or the end of a gene or whatever. Okay, and and then you can tell where the exons are, the introns are, what the amino acid sequence of a protein is, and so on. And this is the second point. So by knowing the sequence of a gene, for example, you know what the sequence of the amino acids in a protein is, and you can predict the structure and function of this protein. So you can tell a lot of things. So I know you're all interested in medicine. You want all to be doctors because of the prestige and image and whatever. But I truly believe that the future is in bioinformatics, medical bioinformatics, specifically prediction analysis, because we have so much data that we don't know what to do with it, and we need prediction analysis. So anyhow, um, also by knowing what the normal sequence of a gene is or a piece of DNA, whether it is a coding region or a promoter or enhancer or whatever, you can tell what the abnormal sequences. You can identify mutations. By identifying mutations, again, you can tell if this mutation is would cause a production of a defective protein that would cause a certain disease or not. Okay. In addition, we all have different DNA sequences, right? So we are similar by 99.9%, but we have this 0.01 0.01% or 0.1% that we're different at, like single nucleotide polymorphisms, for example. And these can increase the variation among human beings. Okay, and that would affect health and disease. It would affect the susceptibility, susceptibility of a person to develop a certain condition, whether it is cancer or heart disease or whatever. Okay, you can also so you can predict again disease susceptibility, and the other thing is that you can use it in evolutionary sciences as well. That is the relationship, the connection between different organisms. You can use it to learn about human migration throughout history. Now, this is a quick history. First, it started with the smaller organisms, smaller viruses and prokaryotes. Then mitochondrial DNA, human mitochondrial DNA was sequenced. Then the yeast genome was done, etc. And in 1990, the Human Genome Project started. Basically, it was an initiative to sequence the whole human genome that is composed of 3 billion base pairs. Okay, and about $3 billion were spent on this project. 7 different countries got involved, and in about 2006 they announced the completion of the human genome sequencing of the human genome, and that was really a huge accomplishment. And I remember that moment, and it was almost as they described it as like the first time man landed on the moon, knowing the book of secrets. So this is just a view of different differences in our genomes compared to different organisms like mice and fruit fly and etc. Now I want you to notice something. You can see that there is a little difference between human genome and mouse genome. The number of genes is really similar to each other. It's not 25,000 anymore. It's about 20,500. Same thing in mice. Notice that in C. elegans, it's a roundworm used a lot in research. Number of genes is 19,000, really. So the difference between the genome of this worm with the human genome is really nothing. Okay, yet we are completely different, of course. Okay, you can see yeast and different bacteria. Okay. The size of genome is also different. It's smaller in bacteria and archaea compared to humans. This is humans right here. You can notice that the diversity in protozoans, for example, if you look at different plants and amphibians, they have larger genomes than ours. Okay. So you don't have to memorize all of these information. This is just spices for this lecture. Okay. So let's start with human sequencing of the human genome. The basic technique was based on the use of a substrate known as dideoxyribonucleoside triphosphate. This one right here. So this is the normal substrate of the DNA polymerase. It's the deoxyribonucleoside triphosphate, which has, this is deoxygenated carbon number 2, and this one here has the hydroxyl group, and this is the important one. Why? Because whenever we want to elongate DNA to synthesize it, we add a nucleotide, the 5' end of a nucleotide to this end, to this hydroxyl group right here. Okay? Now, by using a dideoxy, di means 2, okay? It means that there are 2 deoxygenated groups, including this one here at the 3' end of the nucleotide. Now, because this is not a hydroxyl group, no nucleotide can be added to this group right here, meaning that DNA synthesis would stop. It terminates. It doesn't continue anymore. So this is what we do. Okay, you can read this on your own. Idea here, and you can read this. So here's what we do. Remember what I always say, that whenever I talk about DNA molecule, I'm not talking about a single DNA molecule. Rather, I'm talking about thousands to millions of DNA molecules of the same type. Okay, so whenever I want to do DNA sequencing, I need the following. I need the DNA template. Okay, here I'm showing only one strand of the DNA template. Synthesis goes from 5' to 3'. So what do I need? 1. I need the DNA template. That's 1. The DNA that I want to sequence. 2. I need a DNA polymerase to synthesize the DNA. 3. I need the 4 substrates, the deoxy-ATP, deoxy-GTP, deoxy-CTP, and deoxy-TTP. Okay? And I also need a primer. Remember, a DNA polymerase cannot start DNA synthesis unless there is a starting point. So we use a primer. So I have to have some knowledge of the DNA sequence that I want, or the DNA fragment that I want to sequence, that I want to determine what its sequence is. Okay? And the last thing I need is a dideoxynucleotide. Okay, so I'll have 4 different reactions. Each one of them contains the same template, the same primer, the same polymerase, the same substrates, but they differ by having different dideoxynucleotides. So the first one would have a dideoxy-GTP. The second one would have a dideoxy-ATP. The third would have a dideoxy-TTP, and the fourth would have a dideoxy-CTP. Okay, remember, we're talking about thousands to millions of DNA molecules, primers, molecules of DNA polymerase substrates, so everything is in abundance. So I don't want you to say, what if the substrates are done? What if they're all consumed? There is no such thing. Okay, so keep that out of your mind. Hello. So the reaction that contains the tube, the vial that contains the dideoxy-GTP, it's present at low concentrations. That's an important point. Okay, so let's say that I have 1,000 DNA molecules, the template DNA molecules, 1,000. Okay, now the DNA polymerase would start, and each one of them would have a primer attached to it, or hybridized or annealed to it. Now the DNA polymerase would start DNA synthesis. Okay, so it starts right here. T, it adds an A. C, aha, wait a second. There's a C, so it has to add a G, right? There are 2 G's in our reaction. We have a deoxy-GTP and we have a dideoxy-GTP. Now, there is a chance, and since dideoxy-GTP is present at low concentrations, there is a good chance that a deoxy-GTP is added. If a deoxy-GTP is added right here, it means that DNA synthesis can continue, right? But if a dideoxy-GTP is added, it means that there is termination of DNA synthesis. Okay? So let's say I have 1,000 DNA templates. About, let's say, 100 would be terminated, and there's no synthesis anymore. The other 900 DNA templates can continue with the DNA synthesis. So the DNA polymerase would add, here's an A, it would add a T, A, T, G, C, A, T, A, T, C. Aha! Now, either a deoxy-GTP is added or a dideoxy-GTP is added. If a deoxy-GTP is added, means that synthesis would continue. If a dideoxy-GTP is added, synthesis is terminated right here at this point. Okay, so let's say that we have other 100 DNA molecules with synthesis terminated. We have 800. Now we have 800 that can continue, and so on. And we will have another 100 templates with DNA synthesis terminated right here. So we will generate DNA fragments of different lengths, right? Okay, so if we apply to all 4 different reactions, it means that if in the reaction that I have a dideoxy-ATP, synthesis would be terminated right here, or right there, right? So synthesis will be terminated right here, and right there. Well, in the reaction that contains a dideoxy-TTP, whenever there is an A, there is either a deoxy-TTP or a dideoxy-TTP. If there is a dideoxy-TTP, it means that synthesis would be terminated right here, or right there. Okay, so in all of these reactions, we're going to generate different fragments of different lengths, and they can differ by only 1 nucleotide, right? So in this reaction, it's terminated right here. In this reaction, it's terminated at the 2nd position, and so on. This one right here is terminated at the 3rd position, and so on. We take all of these reactions and we put them in different wells. So this is the well that indicates that synthesis is terminated at the A. This is the well where synthesis is terminated at a T. Termination here is at a C, and termination here is at a G. Okay. Now, of course, the smaller DNA fragments would migrate faster than the larger ones, right? And the separation is so it has very high resolution. Separation can be based on one nucleotide. So this is really how the resolution is so high. So here we have this fragment. This is the shortest one right here. Now, since it appears in this lane, I know that it's terminated at A. This one here is the second. Well, this is the let's say, okay, uh, whatever long DNA. This is longer by one nucleotide. This is longer by another nucleotide. This is longer by one nucleotide versus this, and so on. So I know based on the location of the fragments what the nucleotide is. I know it's A, T, G, T, okay, C, A, G, and so on. So I go like this, A, T, G, T, and here it's the 5' end. Why? Because synthesis starts at the 5' end and it goes in this direction into the 3' end. So I know that this is A, followed by T, then G, then T, and so on. So here what I'm doing is I am reading the newly synthesized DNA. Now I forgot to mention, by the way, that the primer is labeled. Okay? So the bands that I see here, I see them because there is radiation, there is a signal that is coming out of the DNA, and it comes out specifically from the primer itself. Okay, good? Great. Now, so right here we have this gel, okay? So the sequence of the newly synthesized DNA is what? It is right here, it's T, then G, then C, then G, 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 and I'm moving from the 5' end to the 3' end. If I ask you what is the sequence of the DNA template, you're gonna flip it, right? Because it's anti-parallel. So you can start from the top and you can say it's 5', T, T, A, G, A, so T, T, A, G, A, and so on. Okay. Now you can start from this end, by the way, but you have to indicate that you're starting at the 3' end. Okay, great, good, very good. Now, let's say that I should normally have a band. Okay, let's say that I have a gel and I have these two bands right here at the same exact position. So they have the same exact length. How is that possible? Think about it for a second. I'll give you a hint. Our cells are diploid. What do we mean by diploid? It means that we have 2 copies of every chromosome. We have one from our mother, we have one from our father. So there is possibly a variation by one nucleotide between the mother's DNA and the father's DNA in that particular DNA that we are sequencing. So let's say that the DNA polymerase is reading both DNA from that individual, okay? So it reads this DNA A, the other DNA A, T, T, G, G, C, C, etc. But then there is a variation in one nucleotide. In the mother's DNA, it's a T, and the father's DNA, it is an A. Okay, so the DNA polymerase would read both. And remember, we have again large quantity of DNA molecules from the mother, from the father as well. Okay, or from any individual. So the DNA polymerase would read both DNA fragments, adding T here and adding A on the other one, and it would continue with the same exact sequence because there's only variation in one nucleotide. Okay, so we're going to have DNA fragments having the same length in here. So we know that there is polymorphism. Okay? Or it can be a mutation, by the way. It can be a mutation in one of the alleles, mothers or fathers. Now, let's say that normally there should be a band right here, but it's not here. It appears in the other lane. What does that mean? It means that normally there is an A on each allele, A here and A there, but there is a mutation on both chromosomes, on mothers and fathers, or maybe variation, whatever. Okay, so instead of having a in that gene, there is a T. Okay, so right here it means that there is a mutation, and I hope that this is clear. Now, the thing about DNA sequences that I just talked about is that we're using radioactive primers or radioactive labeling of primers, and radioactivity is really harmful for our cells and our DNA. And it is expensive and laborious. It takes a lot of time to do DNA sequencing using radioactivity. So scientists said, and we used to do it manually, reading those gels that I just showed you. Now, the thing is, scientists said, well, why don't we use something that is less harmful? Let's use fluorescence. Okay, fluorescence is sensitive and it's not dangerous. That's one. And they said, well, since we're using fluorescence, why don't we let the computer read our gels? Why do we have to read them ourselves? It takes time. So they said, you know what, instead of labeling primers, let's label the nucleotides themselves, and each nucleotide would give a certain color. Okay, so we're going to have the DNA template. We're going to have a primer that is not labeled. Okay, we're going to have the 4 nucleotides, the 4 substrates with the DNA polymerase, and we're going to have in the same reaction the 4 dideoxynucleotides as well. Why? Remember, I can differentiate these nucleotides from each other because each one of them gives a different fluorescent signal. And we allow DNA synthesis to continue. So the thing is that it's the same exact thing, except that you're going to have termination at every single nucleotide. Okay, and we will have a detector reading or analyzing the signal that comes out from each band, and each band would give a specific color depending on which dideoxynucleotide is added to terminate that DNA synthesis. Okay, and they will have different lengths as well. I'm going to give you animation videos. You can watch them in order to understand them better. Now, the computer or the software gives us these signals in the form of peaks. So it would look like this. So you can see that there's yellow signal followed by yellow signal. So you know that it is the computer reads C, C, G, G, A, G, C, whatever, depending on what the color is or what the signal is. Okay? Now, the thing is, what if there is a variation? How would that look like? What if there is a mutation? How would that look like? Well, let's say that normally we have these peaks. Okay, so this is read as C, followed by T, followed by A, followed by C. Notice here we have 2 peaks that overlap. Okay, the computer would say, aha, wait a second. There is something in here. We have 2 peaks that have similar height. What does that mean? It means that there is a polymorphism. There is a variation. So remember that these signals come from both mother's DNA and father's DNA. Okay? But if it gets to a certain position and it reads a C here, let's say that the mother's DNA is C and the father's DNA is G, the DNA polymerase would read both, and it would add to a portion of DNA fragments that are synthesized. It would add a C here, and it would add a G on the other one, right? And both of them would be almost equally the same in terms of amount. So the computer would read both the same thing. So we would say, aha, we have 2 peaks. It means that we have a variation. Okay, we have a difference in the nucleotide at this particular position. So think about it. What if there is a mutation on both genes, on both alleles? What if instead of having G on one allele, G on the other allele, we have C and C? How would that look like? Well, compared to what normally it should appear, a single peak representing one single nucleotide, we're going to have a single peak as well, representing one nucleotide, but it's going to be different than what normally exists. Okay? And I hope that this is clear as well. Now, the thing is, all of these techniques that I just talked about are, again, laborious. Scientists keep on thinking of improving technologies. And they said, you know what, I'm doing it one nucleotide at a time, and it's taking a lot of time. So why don't I do something different? And they came up with different technologies, and I'm gonna only talk about one of them. And collectively, they are called next generation sequencing, NGS. And that means really fast sequencing of DNA. And this is how it's done. It's all computerized, it's all automized, and it's really genius. And this is how it's done. And since we have, by the way, the Human Genome Project completed, we can always refer to that database. So what we do is the following. We take a whole DNA from a cell, the whole genome, and we cut it into different fragments, random fragments. Okay, I don't care about endonuclease, restricted endonuclease is cutting a certain position. No, 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 it's just random fragmentation of DNA. Then what is done is that DNA adapters are added to the ends of these DNA fragments. How? Well, enzymatic reactions, okay? We're not going to talk about details. So adapters are added. What are these adapters? These adapters are added for two purposes. One of them is to allow for these DNA fragments to attach to a platform, to a solid surface. That's one. And two, so that they can act as binding sites of primers, the same ones that we talked about before for DNA synthesis. And then DNA synthesis starts. The thing is, okay, it starts like this. Special nucleotides are added, okay? These nucleotides have two properties. One of them is that once one of them is added, the subsequent one, the next one, is not added unless the nucleotide, the first nucleotide that is added, is activated. It's modified so that to allow the second one to be added. Two, the second property of these nucleotides is that, so they are not dideoxynucleotides, okay? They are special nucleotides. The other property of these nucleotides is that they can fluoresce, they give a signal. And this signal can be detected with a special camera. Okay? So here's the step. So we add these nucleotides, with each one of them having different colors. So the A would give a color that is different than the C, and the C is different than the G and the T, and so on. A single nucleotide is added to the synthesized DNA. Now, the subsequent one is not added unless this nucleotide is modified. So now, when it's modified, it's activated, it gives a color, and this color is detected. And the other thing is that the new nucleotide can then be added. Again, this new nucleotide, once it's added, okay, it has to be modified the same exact way so that it can be detected and a new nucleotide can be added, and so on. So here we're not talking about 100 DNA fragments added to this solid support, this solid platform. We are talking about billions of DNA fragments added. So it would look something like this. Okay, we would have all of these different spots. Each DNA is synthesized. Okay, and each one you have a different nucleotide that is added, and each spot would generate a different color than the other ones because they are different fragments. So let's say that we have this fragment right here. Okay, so a G is added and then it gets activated. It gives a color that is yellow, and it's detected and then it's activated so that we can add another nucleotide. So this is round 2. The other nucleotide is added. It's a C. It gives a different color once activated. Then we can add a third one and a fourth one and a fifth one, and you can imagine how fast this is because these are enzymatic reactions, and enzymatic reactions are really fast. So it's gonna look like fireworks, by the way, really fireworks, and I'm gonna show you that. So we're gonna have different clusters. Each cluster has a certain DNA fragment, and each DNA fragment can be synthesized. All are done at the same time. Okay. So it's going to be look like this, by the way. This is just 20 micron, really small. I'm really small. This is only part of that. This is small part, by the way, of what it looks like. All of these are different DNA fragments that are synthesized all at the same time. Each one of them produces a different color in less than a millisecond. So we're talking about microseconds. And the camera is so fast, it detects all of these colors all at the same time. So this is how scary the technology is. And this is done really quickly. Remember how I told you that the Human Genome Project started in 1990 and it ended in 2006. And in fact, it did not end in 2006, but they had to announce that it was done in 2006 because of political reasons. In fact, it was done later on, by the way. Okay, so it took them about 16 years to finish it, more than that. Well, and it cost about $3 billion. Well, nowadays, we can sequence a human genome in about, for one individual, in about 24 hours. And this is done in about, for about $500. So what used to cost $3 billion, now it costs about $500. What used to be finished in 16 years, now it can be done in a day. This is really scary. So I'm gonna leave you with this video of what I've just talked about. I want to warn you that this video contains a lot of details. I don't want you to know the details. Um, just the basic idea of how sequencing is done, okay? So don't worry about the details. So let's start, and I'll let you go through it. Okay, so here we have the adapters are added, and these adapters allow for the attachment of DNA fragments into the solid surface, and they provide a location whereby primers can bind to, and the DNA polymerase can start with the DNA synthesis. Look at the fireworks. I love it. Now, this is now all details. Now what they're doing is they're trying to read or synthesize or sequence the other strands. So we can have sequencing of both strands done at the same time. Okay. This is the future, guys. Bioinformatics, prediction, big data analysis. This is the future. I hope you have enjoyed it.